will cause some changes in endothelial function that eventually leading to a foam cell development or early atheroma. This sometimes goes on to become vulnerable plaques, and sometimes it breaks open, causing thrombosis and causing heart attack or ischemic stroke. And some of these plaques can sometimes heal, become a stabilized plaques. However, we are still really at the process of understanding the detailed mechanisms for some of these processes, especially vulnerable plaques. So why biomedical engineers such as me are interested in atherosclerotic questions? The reason is shown in this slide. If you look at where atherosclerosis occurs, both in humans and animal models, it's been well known that it occurs in specific places, both branching points of the carotid artery or curved end branching points in the coronary arteries and iliac branches. And the same occurs in both animal models as well, especially in the aortic arch, in the lesser curvature regions of the aortic arch. These are all the regions that are associated with hemodynamic disturbances. So it has been proposed that hemodynamic disturbance in these places might be associated with atherosclerosis. But whether flow disturbance per se could actually cause atherosclerosis or not was unknown. So this further shows the relationship between bad flow or disturbed flow and atherosclerosis development. So this is a human carotid artery, and as you will see, here is the plaque. Oops. And you will see the plaque in this area. Well, I guess it doesn't like me to use this one. So the plaque occurs in this corner area, and that corresponds to exactly this area where you will see these swirling patterns of flow. And this is really showing the good correlation between flow disturbance and potentially atherosclerosis areas. So before I go further, I'll just give you one more introduction to the talk. So again, endothelial cells, this shows arterial wall, and within the arterial wall there are two major mechanical forces. One is blood pressure caused by the volume of the blood, and the other force that is well known is shear stress. This is a viscous fluid going across endothelium, dragging across the cell surface, and that's shear stress. So one of the bottom line message is that if you have unidirectional high level of shear, that's good. That protects endothelial layer, that protects against the disease. But if you have a shear stress that changes this direction at a low speed, then that usually is associated with disease development. And again, I will focus today on how endothelial cells that is directly regulated by shear stress plays a role in this process. So again, when you have shear stress, endothelial cells recognize that force using a various mechanosensors. There are almost 20 different mechanosensors, and some of them are at the cell surface, including glycocalyx. Some of them are at the cell-cell junction, including PCAM1 and VE-caterin, and the other exists at the bottom of the cells, such as integrins, and cytoskeleton also serves as potential mechanosensors. And once endothelial cells recognize these different types of shear stress, then they react depending upon what kind of shear stress they saw. So this is really a summary of a lot of work done by a lot of people, including Larry McIntyre and Bob Narum on our campus, where most of these lessons were actually learned from in vitro studies, where if endothelial cells are exposed to a stable unidirectional flow, mechanosensors then recognize it, and then that cause cell signaling pathways, including PI3 kinase AKT pathway, protein kinase A pathway, and MAP kinase pathways. These then lead to a subsequent change in eNAS activation to produce nitric oxide production, but in more chronic terms, they will then lead to changes in gene expression, such as increased expression of KLF2, NOLF2, or endothelial nitric oxide synthase eNAS is known to occur. All these changes then lead to subsequently changes in many anti-atherogenic responses. So, for example, shear stress, unidirectional laminar shear stress inhibits apoptosis, proliferation, inflammation, and thrombosis. In contrast, when endothelial cells are exposed to flow disturbance or oscillatory shear stress, such as in these areas of the branching points, endothelial cells sense that change, then it activates a different cell signaling pathways, including PKC zeta, ASK1, and Jun kinase. These then lead to subsequently activation of many stress-related responses that lead to activation of ROS and reactive oxygen species and nitrogen species increase. These then further lead to changes in gene expression, including NF-kappa B and bone morphogenic proteins. These changes eventually lead to changes that are consistent to pro-atherogenic responses, including activation of apoptosis, inflammation, proliferation, and thrombosis. So these were really lessons learned from in vitro studies. However, whether these changes are really responsible for atherosclerosis development or not was really unclear. So my question was really, does flow disturbance or disturbed flow really induce atherosclerosis in vivo? Our hypothesis has been that disturbed flow, characterized by low and oscillating flow, regulates endothelial gene expression, priming endothelial cells for atherosclerosis in the presence of additional risk factors, such as hypercholesterolemia. So to test this hypothesis, we developed an animal model. This is a partial carotid ligation model of atherosclerosis using mouse. So mouse, as well as humans, we have two carotid arteries, left and right. And one of the reasons that we chose this area is because both these common carotid artery regions, right common carotid artery, left common carotid artery, they are straight, they are always exposed to unidirectional flow unless we do something, and they never develop atherosclerosis. So we wanted to change flow in one side, and we can use the other side as a control side. So to change flow in one side without really touching this area, we did a surgery called partial ligation by ligating 3 out of 4 small branches, and we left one branch open, superior thyroid artery. This then led to following changes. If you look at this ultrasound Doppler measurement before ligation in the left side, you can see the signal always goes in each cardiac cycle one way. That meaning there is a pulsatile unidirectional flow. However, after the ligation, then you can see the signal is actually reverting. So that means flow is flipped in each cardiac cycle. And so we did a computational modeling study with Don Giddens group, and this is what we found. And we found that flow goes always unidirectionally in a pulsatile manner in the right side, but after the surgery in the left side, flow goes only briefly forward, but then it goes backward throughout the cardiac cycle. So we were able to generate a low and oscillating flow that mimics what happens in the natural areas. So using this method, our question was, can this flow disturbance cause atherosclerosis? To test this, we took a mouse called ApoE knockout mice. They actually increase cholesterol levels very high upon high cholesterol diet. So we did the partial ligation surgery using ApoE knockout mice, and then we gave them a McDonald's diet, really high-fat diet. It's really a super Mac diet for 2 weeks. And this is what happens. in the left side, in the entire length of the carotid artery develops atherosclerosis, while the right side, the control side, where they were exposed to good flow, they are prevented from atherosclerosis. So in the same animal, this is what's happening. And if you look at the details in the cross-section here in a time-dependent manner, this is what you see. In the left side, where flow is bad, 1 week, 2 week, you can already see this red staining, meaning you have atherosclerosis already developing in 2 weeks. By 3 weeks, it becomes bigger. By 4 weeks, it's almost closely closed off. And again, this also shows additional signs of advanced lesions, intraplaque neovascularization, and a lot of cholesterol cleft. At the same time, if you look at the control side, they are clean, no atherosclerosis. So we have been using this model to really understand how atherosclerosis occurs, and for the first time we were able to demonstrate that yes, flow disturbance causes atherosclerosis. And this is using the same model in 2 weeks by ultrasound. In the right side, you can see in the entire length of the right common carotid artery, the flow is very clean, there's no disease. In the left side, again, you can see plaque occurring in the entire wall here with very narrow lumen. And so using this model, we have been studying how flow disturbance causes atherosclerosis over time. So this is a summary of many papers. So as soon as we do the partial carotid ligation, flow disturbance occurs, and this is sensed by endothelium. This then leads to changes in eNOS uncoupling and activation of NADPH oxidases to produce reactive oxygen species. And by 1 week, this then leads to a endothelial dysfunction. By 2 weeks, it induces atheroma formation, and by 1 month, we can see advanced lesion forming. And by about 4 days and on, we can also see immune cells, leukocytes, infiltrating into the arterial wall. So the next question that we really wanted to test was that what are the mechanisms that a simple flow change could lead to all these atherosclerotic responses. So our hypothesis has been that it must be changes in genes, mechanosensitive genes, at the level of messenger RNA, microRNA, and epigenetic changes and metabolite changes. So to test this hypothesis, this is what we did. And actually, we were very lucky that we chose this a straight part of the carotid artery, because it's like a long pipe. So after we do partial ligation surgery, we can cut it out and we can actually simply flush it. And believe it or not, this was such a simple technique that gives us almost pure endothelial genes. And this technique, a very simple technique but very effective technique, allowed us to do many omics-based studies. So first study that we did was using endothelial enriched genes, total RNA. We were able to do transcriptome studies by doing messenger RNA array studies. And then we did a microRNA array studies to figure out which microRNAs go up or down in a genome-wide manner. And then we then obtained endothelial genomic DNA from these animals, and we were able to do, again, a genome-wide epigenetic DNA methylome studies using a technique called reduced representation bisulfite sequencing and deep sequencing. This allowed us to look at the entire genomic picture of what happens by flow conditions. And we recently have completed this metabolomic studies where we can collect 50 microliter of blood, and then we give it to one of our colleagues at Emory, Dean Jones, and his lab can do metabolomic studies by mass spec, which then tells us more than 10,000 changes in metabolites all at once. And this allowed us to look at the entire metabolomics changes as well. So what we have so far is we have omics data coming from messenger RNA, microRNA, epigenomic DNA methylome, and metabolomics change. Again, these are huge amount of data, and we are now relying on systems biological approaches to get a better handle on our own data to get better ideas of how we go about using these data. So at the end of the day, we look at the pathways, we look at specific candidate mechanosensitive genes, and then we validate them, and we do functional studies. And from these type of studies, we not only understand the disease mechanisms, but also we can actually develop therapeutics. And one of the examples is shown in this slide, where here is a atherosclerosis, and if we block one mechanosensitive gene, this is what happens. We can prevent atherosclerosis as an example. So I'll give you a little more examples of some of these approaches. So, and also another thing we just completed that we are still working on is we have completed the RNA sequencing from our data, and this is to understand a new area called long non-coding RNAs. So this was our first transcriptome data, where we did a messenger RNA array studies using endothelial genes, and we found that flow disturbance changes gene expression for more than 500 genes. And of these, we followed some of the genes that we identified from here. And one example is DNA methyltransferase 1, or DNMT1. Now, the reason that we chose to look at DNMT1 is because this DNA methyltransferase 1 is an epigenetic master regulator. What it does is that it puts a methyl group on cytosine when it is in CG sequence. And what the importance of this gene is that if this gene is involved in many diseases, including cancer, such as acute myeloid leukemia and myelodysplastic syndrome, and there has been a specific drug that inhibits DNMT, and that drug is called 5-ASA-2-deoxycytidine. And so because of this type of work, we actually studied further whether this DNMT1 plays an important role in atherosclerosis. And at the time, there was nothing known about the DNMT1 in atherosclerosis or endothelial dysfunction. So the first was to really confirm our finding whether DNMT1 gene goes up and at the gene and protein levels. And here is immunostaining showing that the DNMT1 protein level goes up in the flow disturbed LCA area compared to the control side. And these are shams showing no signs of DNMT1 protein expression. And this is not only in the flow disturbed area caused by surgery, but in naturally flow disturbed areas such as aortic arch. You can see in the lesser curvature area where flow is bad, you will see a lot of red staining. That means DNMT1 is very high in flow disturbed areas, but not in the good flow region called greater curvature area, such as here. So next it was that we then tested whether blocking DNMT1 has any effect on atherosclerosis. Can we prevent atherosclerosis? So to test it, we treated animal with 5-ASA drug in a dose-dependent manner, and this is our control. In the left side versus right side, you will see a lot of atherosclerosis, and if we treat them with 5-ASA drug in a dose-dependent manner, you can see 5-ASA prevent atherosclerosis significantly in our partial ligation model. We also did additional studies where we didn't do any surgery, and we just gave them Western diet for 2 months. And if we do that, then they develop atherosclerosis naturally in these branching point regions of aortic arch. And that also, this chronic development of atherosclerosis can also be prevented if we treat them with the 5-ASA. So we now have idea that 5-ASA or DNMT plays a very important role. So then we wanted to understand what is really the mechanism, how does it occur. So to get that, what we did was we used this RRBS DNA methylome data and compared that to a transcriptome data, because we got both data sets, DNA methylome data and gene expression data from the same animals. So we then compared to see whether DNA methylation pattern did anything matches any gene expression patterns. The basic idea is this: when genes, especially at the promoter regions, is DNA methylated, then it tends to prevent its gene expression. So if we, by comparing hypermethylation of the promoter regions of each gene and its gene expression, we can actually figure out which gene is behaving in a consistent manner. And we also used 5-ASA data as well in addition to understand it. So by going through this systematic systems biological approaches, we then were able to find some genes. Actually, we were surprised that we only found 11 flow-sensitive genes that are regulated in this manner by DNA methylation and gene expression pattern in a 5-ASA dependent manner. And these are shown in here. So these were the 11 genes that we identified from a genome-wide, completely unbiased approaches. And these were, and another important thing was that out of 11 genes, half of them, including HOXA5 and KLF3, contains a cyclic AMP response element. So from our completely unbiased approach, we found that cyclic AMP response element genes are somehow regulated by this DNA methylome dependent manner. And that's something that we are actually actively following to figure out what this means and how we can actually get a better understanding. So the next topic that I'm going to cover is microRNA. MicroRNA is a non-coding small RNA, and when it's mature, it's about 22 nucleotides, and I'm sure many of you have heard about it. If you haven't, think of it as a cousin of siRNA, small siRNA that behaves like siRNA, and this is how it works. So it is made in the nucleus through like any other genes, through transcription, and this is initially made into pri-microRNA. It's a longer sequence, and this pri-microRNA can be is then processed by enzyme called DROSHA and DGCL8 complex, and this is then made into a premer. Once you have smaller premer, it goes out into the cytosol through exportin 5, and then it binds to, and then it uses dicer, which further processes pre-microRNA into mature form, and then it puts into a RISC complex. Once it goes into RISC complex with the help of AGO2 proteins or AGO proteins, then it does following things. Once it binds to specific genes, and then it degrades. This is one of the best things that happens. Also, or it prevents translation, or sometimes it also induces de-adenylation. So all in all, these result in a decreased or a degradation of specific gene targets. And there have been many shear-sensitive microRNAs reported, but at the time when we first reported, there was no reports regarding whether any of these mechanosensitive microRNAs play a role in atherosclerosis. So this was our initial array data showing a lot of mechanosensitive microRNAs, and we have validated many of them. And another thing that we did was that in each of our studies, actually we do both not only in vivo studies, but concomitant in vitro studies where we collect endothelial cells, we culture them under either laminar shear stress conditions that simulate good, stable unidirectional flow, or oscillatory shear that simulates flow disturbance. So this is how it looks like. When you culture endothelial cells on the laminar shear, they align to the direction of the flow. In oscillatory shear manner, they show a cobblestone, no particular directions, and then we can actually confirm what we saw in vivo, in vitro conditions as well. So from this type of work, we have selected additional microRNAs that we wanted to study, and one of them is called microRNA 712. So this microRNA goes up by bad flow. And long story short, what we identified was that this microRNA targets this one called TIMP3, tissue inhibitor of metalloproteinases, or MMPs. And one of the reason that this may be very important is because TIMP3 is an endogenous inhibitor of MMPs and atoms. And TIMP3 is well known, as well as MMPs are well known in atherosclerosis. And one of the critical things is that TIMP3 is lost in many chronic disease conditions, including atherosclerosis, abdominal aortic aneurysm, and cancer metastasis and heart failure. So, however, why TIMP3 is lost in many of these chronic conditions was unknown until we actually provided this first evidence that this microRNA is actually one of the upstream regulator of the TIMP3. So I'm going to skip some of the details. So then what we did was we then studied how what this Myo712 is doing in endothelial cells. So first thing that we identified was that microRNA 712 induce inflammation, such as in this in vitro experiment, where we can see if we incubate cells, endothelial cells with Myo712, as opposed to the control, inflammation is occurring as measured by monocyte adhesion to endothelial cells. Another thing that happens is that the permeability goes up. So if we treat endothelial cells with Myo712, permeability to albumin goes up. And this is mediated by TIMP3, because if we overexpress TIMP3, we can actually bring back the increased permeability. So TIMP3 plays an important role in this Myo712 pathway. So another thing was that we then looked at in vivo what happens. How does Myo712 work in an in vivo condition? So again, this is our hypothesis. Flow disturbance increases Myo712, and if you have increased Myo712, then this downregulates TIMP3. And so this is a protein staining showing in our control right side versus left side. As you can see, the red staining showing TIMP3 protein level, which is highly expressed in the healthy arteries, but in response to bad flow, they lose TIMP3. This lost TIMP3 can be rescued if we treat mice with anti-Myo712. So this again shows that our hypothesis is correct. And once you lose TIMP3, as I told you just previously, TIMP3 is the endogenous inhibitor of MMPs or atoms. If you lose TIMP3, MMP goes haywire. So we expect that once you lose TIMP3, we will see very high levels of MMPs and atom activities. And I will show you some data using this gelatinase activity assay, as well as staining for atom activity assay. So again, this is MMP activity measured in vivo, and this is atom activity, again, used in our in vivo samples. So to measure MMP activity, we used a fluorescently labeled gelatin. And if you see a lot of greens, that means MMP activity is high in LCA, flow disturbed side, compared to the control side. But if we inject mice with anti-Myo712, we can actually prevent MMP activation. Again, suggesting the important role of MMPs, the Myo712. So then we measured atom activity by using versicon fragmentation assays. And you can see in the control there is not much signal going on. But in the LCA, you will see a lot of red staining. I don't know if you can see it. That means Shedase activity has increased. And this increased atom activity is again prevented if we treat mice with anti-Myo712. So finally, then we wanted to see whether this anti-Myo712 actually can prevent atherosclerosis development. So again, here's our control. You can see in the left side by flow disturbance, the entire length of the left common carotid artery develops atherosclerosis. And this atherosclerosis can be prevented if we treat mice with anti-Myo712. So we gave anti-Myo twice a week at 5 milligram per kilogram dose through subcutaneous injection. And we were able to demonstrate that anti-Myo712 is a very potent anti-atherogenic therapy. And again, we also treated chronic animals without doing any surgery, just giving them high-fat diet. So in 2 months, they develop atherosclerosis, especially in the aortic arch areas, without any surgery. And this kind of chronic atherosclerosis development can also be inhibited if we treat mice with anti-MIO-712. So in summary, what we showed in that study was that in endothelial cells, when they are exposed to flow disturbance, this then leads to activation of MIO-712. Now, there are a very novel pathway that we also described in this paper, but due to time, I'm going to skip that part. Anyways, through a XRN1 dependent pathway, we were able to show that MIO-712 goes up in flow-disturbed endothelium. This then goes, some of them remains in endothelial cells, some of them goes out into circulation, as well as some of them goes into a medial smooth muscle cells where they really have impact as well. In endothelial cells, MIO-712 decreases TIMP3. Once you lose TIMP3, MMPs and atoms go up, and this then leads to changes in many changes, including extracellular matrix fragmentation and permeability increases. And some of these are also this MIO-712 going out to circulation acts on circulating leukocytes, activating them and increasing the inflammation. So they bring leukocytes such as monocytes into the subendothelial layer, which then becomes foam cells and subsequent atherosclerosis development. Now, after we did this, we also looked at another disease called AAA, or abdominal aortic aneurysm in mice. And this is our control experiment where you can see, here are the kidneys for your reference. This is AAA developing in mice. And this type of AAA can also be prevented if we treat them with the anti-MIO-712. So these type of studies demonstrate the importance of anti-MIO-712 or MIO-712 activating MMPs, leading to atherosclerosis and AAA development. So now, so far, I have been using anti-MIO-712 by subcutaneously injecting it, a naked form of anti-MIO-712. We just dissolve it in PBS. We just stick it in underneath the skin, and it goes everywhere. And it affects every cell, actually. And so not only it affected endothelial microRNA 712, but it also affected many other cells, so some of which may not be actually so good. So we wanted to develop additional mechanism whereby we can introduce MIO-712 only to areas where they are inflamed. If we could achieve it, I think this could actually really improve our therapeutic potentials. So this study, what we call Nanomedicine Study, was done in collaboration with my colleague, Kathy Ferrara at UC Davis. And this was just published in ACS Nano. So here we are targeting VCAM1 as a marker of inflamed endothelium. And then we then hope to put our therapies inside the nanoparticle. So this is how our lipid nanoparticle look like. Through a really elaborate sequential pathways, synthetic pathways, we developed this lipid nanoparticle. So within this lipid nanoparticle, anti-MIO is inside and is surrounded by positively charged cationic lipids, surrounded by a neutral lipids outside, and then it has pegs to increase the circulation time, and some pegs also have this peptide moiety. This peptide is VCAM1 targeting peptide. And so then what we did was then we injected this nanoparticle containing VCAM1 targeting moiety as well as anti-MIO-712. Again, before I show you that data, here's our control. So we wanted to see where VCAM1 was expressed on endothelial surface. So both in LCA, where flow is disturbed, we can see a lot of red staining, meaning VCAM1 is high compared to RCA. And also in aortic arch, without any surgery, in a naturally flow disturbed areas, we can see a lot of VCAM1 expressed in flow disturbed areas, but not in the greater curvature area where flow is good. So we know VCAM1 is expressed in flow disturbed regions and pro-atherogenic regions, and if we inject microRNA containing nanoparticle, it goes and binds to only LCA area where flow is bad, or lesser curvature area in the aortic arch, showing that our nanoparticle only goes to places where VCAM1 is high. And if we don't put the targeting peptide, it does not bind. So it shows us that our targeting strategy is very specific, and it binds to endothelial surface. And we were actually surprised that this nanoparticle binds to endothelium, it gets inside the endothelial cells, but it goes no further. So we don't know why, but it gets stuck in the endothelial layer. And I'm going to skip the in vitro data, but we were able to show that in that in the in vitro condition, it goes into the cells, and it works really specifically to block Meo712. This is in vivo data. So what we did was we did a partial ligation surgery to induce Meo712 expression in the LCA. And then we injected mice with this lipid particle with the VHPK, the VCAM1 targeting peptide. And if it works, this is what should happen. It goes into endothelial cells, and it should prevent the increase in Meo712. And that's what we really saw. And as not only Meo712 level changed, its downstream gene target called TIMP3 actually gets rescued. As I told you, TIMP3 level drops in flow disturbed areas, and this can be rescued by treating animals with a VCAM1 targeting anti-Meo712. So this was our final experiment using our nanoparticle. So again, this is our control experiment showing a lot of atherosclerosis in the LCA by following partial ligation, and this atherosclerosis can be prevented if we treat mice with this VCAM1 targeting anti-Meo712, and this does not work if we don't put the targeting peptide in the front. So again, from this experiment, we were able to demonstrate that our anti-Meo712 delivered in VCAM1 targeting nanoparticle goes specifically to endothelial cells and prevents atherosclerosis. And this shows that actually this anti-Meo712 does not have any side effects that as far as we can tell. So we looked at the potential side effects of these nanoparticles. We collected samples from many tissues, and liver, kidney, lung, spleen, thymus, bone marrow, and circulating PBMCs, etc. We don't see any effect of anti-Meo712 in any of the tissues that we looked. So we don't have, so far, we are not seeing any side effects of this anti-Meo712 nanoparticle. We only see its effect in inflamed endothelial cells. So in summary, this is what we have done. We are going with the flow to understand atherosclerosis and to develop a new anti-atherogenic therapies. So in summary, we showed that partial carotid ligation induces flow disturbance, which is sensed by endothelial cells immediately, that lead to changes in genes, pro and anti-atherogenic genes, including Meo712 and DNA methyltransferase 1, that subsequently lead to changes in TIMP3 and MMP activities, as well as some target genes leading to endothelial dysfunction, atherosclerosis formation, and advanced lesion formations. We have also developed a new anti-atherogenic therapies, such as anti-Meo712, as well as we showed the therapeutic effect of 5-azar as an anti-atherogenic therapy. So far, what we have done is that we have taken this omics approach using our partial ligation model. From this, we have really selected a few genes. We still have a lot more to go based on our omics data. And then we will continue to study mechanistic insight as well as new therapeutic development. And we will continue to develop our nanomedicine side. So far, we have been doing really well in terms of preventing atherosclerosis in mice. But that's not my end goal. I think our end goal is somewhere downstream. That is, this is where we want to go. We want to develop a therapy that will be actually useful for humans. And at the beginning, I told you that we have statins, cholesterol-lowering statins, and drug-eluting stents. Despite that, the atherosclerosis is still #1 killer in the entire world. We need a better therapy. And I think what we have done here could actually provide the different therapeutics that are not, that the statins or drug-eluting stents cannot cure. So how do we go from here to here? I think we want to take a route where we are actually beginning to work with bigger animals such as baboons and pigs, and continue to develop a targeted therapeutic deliveries. And these will, I believe, help to take us to the clinical trials. So again, I have to tell you that I have not done anything myself. And these are actually the group of kids, my students and fellows who in the lab who have done all the work, including Rachel here. And here are actually the most important people who have done the work. Sandeep Kumar, currently a research instructor in my lab. Dongju Son, Chanu Kim, Jesse Dunn. Jesse did the epigenetic studies. And my lab manager, Dongwon Kang. And Amir Rezban, who is now assistant professor at Emory Cardiology, have really contributed a lot to this study. And my colleagues at Georgia Tech in this department, Don Giddens, Bob Nerum, Ajit Yoganathan, King Jordan, and Yun Fan really helped a lot. And Kathy Grindling, Bob Taylor, Dean Jones, and Youngmi Go at Emory is my important colleagues, as well as colleagues in UC Davis and Goethe University, Ralph Prentice. And these are my funding sources. Thank you very much for your attention. And if you have any questions, I'll be happy to answer.
That was great. I came in late, but I got most of it. The nano data looked amazing. Can I ask you, I mean, I would still guess most of the particles goes to the spleen and the liver and the kidney, and very little goes to the vascular endothelium. The reason that you don't see any mirror effects in the other organs, I saw that it was normalized to the scrambled peptide target. If you normalize it to the untreated animal, because in those cases, if VCAM1 is not upregulated, you would expect that the scrambled peptide and the actual peptide will give you the same changes.
Right.
So you would not see a difference.
Right.
But if you do it in comparison with an untreated, do you see that?
We have not compared it to untreated, so I don't know the answer.
I mean,
yes.
Going selectively to a vasculature is very difficult in nano.
Right. We were actually very surprised that we didn't see any effect.
Yeah.
I think one potential reason is that because we are actually taking the whole liver or whole tissue.
It's a mass effect, right?
So maybe there may be some little effect that is hidden, but we can't tell. But when you look at the entire liver or entire tissues, or actually when we looked at blood cells, this is actually a cells, and that's where actually we would have seen some effects.
Well, one of the things that we find in our most of the nano stuff is you see very acute inflammatory signature. Almost all nanoparticles, especially if there is a cation, it will trigger systemic cytokine within an hour.
Actually, we measured it, and we haven't seen. So there is a little bit of complementary C5a activation, but in terms of any other inflammatory markers, right? We have measured it, and it didn't change.
So of course, this is really nice, right? Particularly the nice progression. So I guess my question is about the human part, as how the therapy would be used. So do you imagine if you give it to a person, like if it's in the middle of their plaque growing, or at what point will it either stop it in its tracks, or do you think there's any reversal that could happen? I guess that's the first question. And then the second question, is there any crosstalk between the MIR and DNMT1? Like, does one win out over the other? Does one control the other?
So the second question first. So DNMT1 has some controlling site in the 3', its 3' end, that where actually some microRNAs actually control DNMT1 expression. So that's also mechanosensitive microRNA that regulates DNMT1. So there is that connection. In terms of whether we can use this as either therapeutics or just a prevention, so far we have focused on just preventive studies because that's easier to do. And I think, so that's actually, clinically, I think prevention or stopping the disease progression, whatever that is actually a very important aspect of clinical use of this type of work. So I think that regardless of, we don't know therapeutics effect. We haven't really tried. But I think we are, at this point, we are just focusing on this preventive effect first because it's easier to do.
Hey, I had two questions. What happened to mice that are ApoE wild type but ligated? And then, what is the sort of thought as to the de novo methylation versus maintenance methylation roles for DNMT1 and the
DNMT3.
subfamily of DNMT3 in mice?
So DNMT1, which is DNMT1 and DNMT3 are both mechanosensitive. So although we see a lot more DNMT1 regulation, but DNMT3 seems to be, which is involved in the de novo methylation, DNA methylation, is also activated, but to a lesser degree compared to the maintenance methylation regulated by DNMT1.
But 5-AZA would target both.
Well,
so
right. But it turns out that, so in our model, DNMT1 is really the major one that changes. So 5-AZA is actually more selective toward DNMT1 than DNMT3. So while we cannot exclude the role for DNMT3, I think it does have minor role. But the major role seems to come from DNMT1. And what was the first question?
What happens to mice that are
the ApoE, right? So it's a wild-type mice. So far, it required ApoE knockout or LDL receptor knockout, but actually we recently have been able to do it in C57 black wild-type mice by just raising cholesterol levels using something called a PCSK9. So PCSK9 is made from liver, and it goes out into circulation, and it binds to LDL receptor. Actually, it leads to a downregulation of LDL receptor. So if you have high level of PCSK9 through viral infection, etc., then actually you can effectively cause LDL receptor reduction. And if we do that, actually we can use regular mice, and we can cause atherosclerosis.
So no Chick-fil-A, and I'll be okay.
Chick-fil-A may be needed, or if it is not Supermac, it may be diabetic conditions, or so flow alone does not lead to all the way to atherosclerosis. I think flow disturbance is what really sets off a chronic inflammation. But you need additional factors such as high cholesterol or diabetic conditions, etc., to really brings it to atheroma formation. I think you have a question first.
I got actually 2 questions. One goes along with is we're talking about what genes or mRNA is changed with one or the two of the conditions, whether it be hypercholesterolemia or disturbed flow. And the second question is, when do melanocytes enter the vessel wall of your model versus when do you see these changes?
So I'm going to answer the first question, and then I'm going to ask you the second question later. So are you asking whether there are different pools of genes that are regulated by cholesterol versus flow?
Yeah, I mean, do you see?
Right. Actually, we have that data. We're actually writing a manuscript right now where we treated mice with either statin drug or just without statin drug, and we compared which genes in endothelium are regulated by cholesterol-dependent manner as opposed to flow-dependent manner. Actually, we were surprised to find that almost these two pools of genes are completely segregated. So I think this actually really validate some of our hypothesis that these gene targets that are regulated by high cholesterolemia is almost independent of the genes that we are regulating with flow disturbance. So the second question was?
The role of monocytes in this process. When do you see monocytes versus when you see changes in all of these omic kind of parameters?
Right. So monocyte change occurs really rapidly as soon as we do the partial ligation surgery. And so as early as 4 days after our partial ligation surgery, we can see and measure monocyte coming in and infiltrate into the flow disturbed areas by FACS or staining. And so I think probably the real event occurs a lot faster. How monocyte actually gets activated when what we only did was flow change is still completely not understood. But part of the reason is a factor such as microRNA is produced from endothelial cells in a very local area that goes out into circulation and hits leukocytes. And actually, we have shown that our microRNAs actually have that specific direct role, especially on monocytes.
So do you think all these effects are, related to monocyte? Or do you think monocyte recruitment, at least I know in later process steps, they're very important, but in the earlier steps, how important do you think they are?
Oh, it's a central event. So atherosclerosis requires several cell types. It's not just endothelial cells, it's not just leukocytes or smooth muscle cells. They all actually are all involved in developing. And so each cell has its own roles. So without monocyte becoming macrophage and engulfing high cholesterol and fat, you're not going to have atherosclerosis. But that initial event requires endothelial cells to become dysfunctional. And so these are all related events. Nothing just happens by itself. So there was a question.
So is there any evidence that in humans that patients that are more susceptible to arteriosclerosis have low flow?
Well, good example is that if you exercise, so if you go to doctor's office with a problem with the cardiovascular system, what do they tell you? Diet and exercise. And in fact, exercise, especially the aerobic exercise, which increases cardiac output, actually one of the benefit of the increased cardiac output is on improved endothelial function by actually improving flow patterns and decreasing the bad flow. And I think that has a direct effect.
So are there any studies to show that patients that have any genetic defect that would affect flow or any of the genes that you have mentioned are more susceptible?
So actually there are some studies, especially using carotid branches, how your endogenous shape is actually has a genetic effect. That's genetic effect. The natural shape of the curvatures and branching points. So some people are more susceptible because the hemodynamics will be worse in some certain people.
So I guess my question is, what is the link between flow and this transcriptome change? So we know that microRNA and epigenomics change should be a very slow process. What is that here? We cannot see so.
Well, so there are a lot of master switches. There are many transcription factors. As soon as you induce a flow change, within a few hours, you can actually see a dramatic change in gene expression because a lot of transcription factors are changing, and these really drive a lot of gene changes very rapidly. So there are very rapid change in gene expression, and some of them are more sustained. Some of them actually comes on early on, that goes away, and then epigenetic changes. We presume epigenetic changes would be slower, but I wouldn't actually completely assume that is the case, because I think actually epigenetic change may occur actually sooner than a lot later.
And the second question is, what is the phenotype dicer knockout, do they form more atherosclerosis?
I think actually somebody has published that paper, but if you knock out dicer, it changes so many things. So its phenotype is really hard to interpret. Okay, thank you very much.